Welcome to this lecture on the applications of animal biotechnology. Imagine you have a massive manufacturing factory that normally produces standard cars, but you suddenly swap out its instruction manual so it starts producing life-saving medical ambulances instead. In the biological world, living cells act as these incredible microscopic factories, and their DNA serves as the ultimate instruction manual. Today, we are going to explore how science rewrites these manuals to solve massive real-world problems, from producing vital medicines to diagnosing hidden diseases. Now that we understand the basic premise, let us look at how these biological factories operate. Every living cell functions like a highly efficient microscopic factory. The machinery inside these cells is guided by DNA, which serves as the ultimate instruction manual. Animal biotechnology is simply the art and science of rewriting or utilizing these biological manuals. By altering the instructions, we can command these cellular factories to produce entirely new, valuable outputs. Whether it is creating cows that produce medicine in their milk or fixing a broken gene that causes a terrible disease, biotechnology turns biological systems into powerful tools for human and animal welfare. Building on this idea of cellular factories, let us formally define what this field entails. Animal biotechnology refers to the use of scientific and engineering principles to modify living organisms, specifically animals, or their biological systems to produce valuable products or services. One of the most advanced techniques we use is recombinant DNA technology. This is where DNA from one species is carefully extracted and inserted into another. When an animal receives this foreign DNA, it becomes what we call a transgenic organism. This allows scientists to enhance specific animal traits or program them to produce vital pharmaceuticals that we desperately need. Beyond creating transgenic organisms, this technology gives us incredible medical capabilities. Molecular diagnostics is a prime example. Instead of waiting for a patient to show physical symptoms of a disease, we can use genetic material to detect the illness at a fundamental molecular level, long before the patient even feels sick. Furthermore, animal biotechnology includes gene therapy, which is a revolutionary medical approach. If a patient has a genetic disorder caused by a defective gene, scientists can use engineered viral vectors to deliver healthy DNA, effectively replacing or repairing the broken gene. We are literally fixing the instruction manual from the inside out. These advanced techniques might sound like science fiction, but their real-world impact is profound. The importance of animal biotechnology cannot be overstated, as it literally saves millions of lives every single day across the globe. Consider the treatment of diabetes. Before these modern advancements, patients had to rely on insulin extracted from the pancreases of slaughtered pigs and cows. This process was highly inefficient and sometimes caused severe allergic reactions. Today, we use genetically modified bacteria or mammalian cells to produce pure human insulin in massive, perfectly safe quantities. Without these biotechnological applications, modern medicine and targeted genetic therapies would simply cease to exist. To achieve these medical miracles, we often face a significant practical hurdle. To diagnose genetic diseases or clone genes for therapeutic proteins, scientists often start with a microscopic amount of DNA that is far too small to analyze. To solve this, they use a fundamental biotechnological application called polymerase chain reaction, or PCR. PCR acts like a biological photocopier, copying a tiny trace of DNA millions of times. The growth of these DNA molecules follows a strict mathematical progression because every single strand is duplicated in each heating and cooling cycle. Understanding the mathematics behind this amplification is crucial for predicting exactly how much genetic material will be produced. This mathematical progression leads us directly to the core formula used in PCR amplification. The formula is n equals n0 times 2 to the power of n. Let us break down what each symbol means. The variable n represents the final number of DNA molecules produced after the amplification process is complete. The symbol N0 stands for the initial number of DNA molecules we started with in our biological sample. Finally, the variable N represents the number of thermal cycles the PCR machine has successfully completed. Because the DNA doubles every single cycle, the base of our exponent is 2. As the number of cycles increases, the final amount of DNA grows exponentially, allowing us to detect even a single viral particle. Now that we have our formula, let us see this with a concrete example. A veterinarian is testing a dog for a severe genetic disorder, but only manages to extract 5 copies of the target disease gene. The lab needs at least 1 million copies to run a diagnostic test. The PCR machine is set to run for exactly 20 cycles. We substitute our given values into the equation. N equals 5 times 2 to the power of 20. The term 2 to the power of 20 calculates the massive amplification factor, which is 1,048,576. Multiplying this by our initial 5 copies gives a final answer of 5,242,880 copies. Since this is well over 1 million, the amplification is highly successful. Let us look at another scenario where we need to work backward. A lab is screening cattle for a viral pathogen. The machine requires exactly 32,768 copies of viral DNA to trigger an alarm. The initial sample contains only 8 copies. How many cycles must the machine run? We set up our equation. 32,768 equals 8 times 2 to the power of n. To isolate the exponential term, we divide both sides by 8, giving us 4,096 equals 2 to the power of n. By recognizing mathematical patterns, we know 4,096 is 2 raised to the 12th power. Since the bases are identical, the exponents must be equal. The machine must run exactly 12 cycles to reach the detection threshold. Now that we have used this formula to solve real laboratory problems, let us understand exactly where it comes from. We are setting out to derive the mathematical progression of PCR. We start with an initial number of DNA strands, which we call N0. During the first heating and cooling cycle, every single strand is duplicated. So after cycle 1, the number of strands is N0 times 2. In the second cycle, this entire new population of DNA duplicates again. So after cycle 2, we multiply the previous amount by 2, which gives us N0 times 2 squared. You can start to see the intuition here. Every cycle adds another multiplication by 2, building the foundation for exponential growth. Continuing from our setup, let us finalize this derivation. Following the exact same biological pattern, after the third cycle, the total number of strands becomes N0 times 2 to the third power. If we generalize this process for any arbitrary number of cycles, which we call n, we are simply multiplying the initial amount by 2 n times in a row. Mathematically, repeated multiplication of the same base is expressed as an exponent. Therefore, the final number of molecules, n, is exactly equal to n0 times 2 to the power of n. This simple yet powerful derivation proves why we can predict biological amplification with absolute mathematical certainty. So, we have proven the mathematical formula. This brings us to a visual representation of what is actually happening inside the PCR machine. On the screen, you are looking at a graph of exponential DNA amplification. The horizontal axis represents the number of PCR cycles, while the vertical axis tracks the number of DNA molecules. Notice the smooth, upward curving line. It starts slowly at one molecule, but as we move to cycle 1, 2, and 3, the number jumps to 2, 4, and 8. Below the graph, the schematic shows the physical DNA double helices splitting and copying. This diagram perfectly illustrates how quickly exponential growth escalates, allowing us to turn a microscopic trace of DNA into a massive, analyzable sample. We have covered a lot of ground today, so let us review the key takeaways. First, animal biotechnology fundamentally transforms our understanding of genetics, allowing us to treat biological cells as programmable factories to produce essential medicines and diagnose hidden diseases. Second, we learned that techniques like PCR rely on the exponential amplification of DNA, governed by the formula n equals n0 times 2 to the power of n. Finally, we saw how mathematical models allow scientists to predict and control these biological processes with absolute certainty. By mastering these concepts, you are now equipped to understand how biotechnology solves some of the most pressing medical and agricultural challenges facing the world today. Great job!